Okay, so we are now into chapter 11, and we're just going to speed on through this. So, this chapter is all about the small molecule transport. So, pretty much what causes or what aids in the transportation of ions, small molecules, across a lipid membrane. Okay, so just knowing that certain things that we are going to be seeing in this chapter are the inorganic ions. So there's your sodium, your potassium, your magnesium, calcium, even the hydrogens are all going to be found in this process. Now, given that we're talking about lipid membrane, also understand that we are talking about concentration gradient. We're talking about diffusion. We're talking about facilitated diffusion. Osmosis, again, is the diffusion of water specifically, which we will touch on, but mainly we want to focus on the sodium and potassium behaviors here. Okay. So we know that we are working with a lipid bilayer. Now, with this lipid bilayer, certain things can pass through fairly easily and certain things cannot. We know that non polar molecules, so hydrophobic molecules, can pass through the lipid membrane fairly easily. One, they're being repelled by water, so that aids in the movement across the membrane. But number two, just know that, again, with a whole bunch of fatty acid tails in this region right here, so on both sides, and then you have your head, on the other side, right? That's to pass this hydrophobic layer will require some sort of energy. Now, it doesn't mean that all polar molecules can't go across it. It just means that they're going to have a much harder time. And depending on how big or how small it is, will determine whether or not they are stuck on the phospholipid heads or they can pass through. So O2, CO2, N2, steroids, and other types of hormones can pass through because, again, it can interact with the hydrophobic layer and get shot out very, very easily. Not really too much of any difficulty there. Now, if we're looking at water molecules, urea, glycerol, and amine groups or ammonia, it depends. Okay. Now, take a look at the arrow. The arrow shows that it's much less in terms of concentration as the hydrophobic molecules. That means some can get past, but a lot of times they're going to get stuck on the phospholipid heads right here. And eventually something else is going to interact with these phospholipid heads where it kind of shoots off the say glycerol that was interacting with the phospholipid heads. Now remember from last chapter, phospholipid heads and even aminos have different types of sugar molecules attached to it. Okay, that was representing a hexagon. I can't draw hexagons right now too well. So you have a lot of sugar molecules on these phospholipid heads. So a lot of times these polar molecules will interact with the sugar molecules first before interacting with the lipids. Now, if we're looking at glucose, sucrose, again, with the idea of the sugar molecules, okay, uh, they're going to be hyper interactive with the oligosaccharides. Some of it might pass through, just ever so small. It might pass through, but most of the time it's going to interact with the surface of the lipid membrane. And then for anything that's super ionically charged, again, the sugar is the first thing it will come in contact with. So most likely it's not going to even pass through the membranes at all. Okay. So here's a permeability of how easily things can pass versus not. So again, just a rough idea that O2, you're able to pass it very easily. H2Os, now we're hitting to not really so much, and then ions at the very end. Okay. Now there are two types of proteins that aids in the transporting of these polar molecules. And we have the transporter protein and then the channel protein. Now, the transporter, again, is essentially your gate or your doorway for polar or soluble molecules to go from one side of the lipid membrane to the other. A channel protein is essentially a tunnel that can allow things to pass through very, very easily. Okay. Now, active transport, same thing with the three types of passive transport plays a role. With active transport, we do want to utilize a couple things. Number one, we want to utilize some form of energy for the active transport. Now, this energy can come in a variety of different methods. It could be a coupling reaction with another reaction outside the cell that allows the gate to kind of conform and open up. It could have a hydrolysis of a nucleotide or a GTP, ATP. It could have some sort of conformational change. Maybe a cyclic AMP comes in and changes it somehow. Or it could be a receptor protein that opens it up and then has some sort of energy requirement for that. Okay. But just remember, for any type of active transport, we're always coming from a concentration of low concentration to an area of high concentration. Okay. For these types of active transport. Again, it requires energy to pump things against their spontaneous movement. So, spontaneous movement going from high to low in case of simple diffusion or passive transport. Remember, we're going from an area of high concentration to an area of low concentration. If you're going against that natural flow, that costs energy, aka active transport. Now, in terms of energy, there's multitudes of types of energies that we can use. We can also use energy that is stored in an electrochemical gradient. So for this case, we're going to be talking a lot about the chemical gradient of the sodium potassium pump and the ability it has on the membrane potential. But just know that depending on what charges are found on what side, or the net charge is found on the side, will represent one side being more positive than the other side. And in relation, it will be more negative. So the outside of the cell is generally going to be more positive. The inside of the cell is going to be more negative in relation to the outside. Okay, it doesn't mean it's fully negative. It's not like a whole bunch of anions are found on the inside of the cell, but it's just a relation status. Okay, so some confirmation about the bilayer right here, or about the gates. We have the outward open, meaning the solute can come in from the outside, lodging inside the transport protein, binding or interacting with something inside of it, causing a conformational change to occlude or close off both sides. And then something happens. Could be a change in net charge. It could be another conformational change. Maybe another molecule is interacting with it, or maybe it's just it within itself intra relation, where the opposite end opens up, allowing the solute to travel straight through. And this is what we call an inward open. And again, it's following the path of diffusion with the concentration gradient. Okay. Now, just know that because of the gates, okay, we are utilizing a protein. Again, proteins are tools for the cell. Now, it's not an enzyme, right? But it still follows the same behavior as an enzymatic graph, which we've studied in the past. This is the Michaelis-Menten plot. So if you take a look at the red, you'll notice that even with a transporter-mediated protein, there is still a Vmax involved. Now, why is that? Well, if we take a look at this protein, it can, for this example, it can only interact with one solute at a time. Now, imagine if we are trying to transport multiple solutes down a concentration gradient and we only have one door opening and closing at a time. It's like a revolving door in a shopping center. You can only have people come in and out every so often, right? It doesn't matter how many people are on the outside, only like 10 people can go through a revolving door at a time. No more than that. That's the maximum amount of people that can travel through. And in this case, for this protein gate right here, this channel, not channel protein, but a protein mediated gate, we can only have one solute going in at a time. Now, it might be able to open and close faster, but we can still only carry as fast as it can open and close one solute. And so there is always going to be some sort of maximum velocity. And vice versa, we can find the Km, your constant, okay, at half Vmax. Now, this is again for a transporter mediated diffusion process. Now, what about a channel protein where we have the lipid bilayer and then we just have a tunnel? Does it matter how fast or how slow uh, is it regulated? In essence, if you have an open road, does it is it regulated? Not really, right? Simple diffusion can travel across an open tunnel as fast or as slow as it needs to. The more or stronger the concentration, right? Or the higher the concentration of the say anion on one side of the membrane. Say that you have a whole bunch of negative charges right here. Let me get my pen out here. Negative charges on this side. You have a whole bunch of negative charges anions right here, right? Very high concentration right here. It doesn't really matter if it's an open tunnel, right? It's just going to go through as fast as it can until it reaches equilibrium. And so the simple diffusion, where we're looking at the channel mediated transport or just regular simple diffusion, is going to travel at a linear slope. Because again, it doesn't rely on an enzyme or a protein to open and close. So there's an unlimited amount. It's solely based on how much concentration there is and how fast it's going to pour through, and that's it. Okay. Now, again, we do have other ways of performing transport, active transport. Again, we have coupled reactions. So, again, using one reaction to run using the energy from one reaction to open and close the transporter here. We can use ATP driven pumps, or we can use the energy from light itself, where light with the UV radiation conforms the change because it absorbs the light energy going to higher oxidation state, which again affects the molecules around it. So there are different types of paths that we can talk about. We can talk about one path, double path, or anti-paths. And again, this is just a review from GenBio, but if we're looking at a singular path where it only affects one ion or one solute, we call this a uniport, right? It travels in one direction. A symport is generally going to be a co-transporter where you need to have both molecules present in order to go through the gate for this case. An antiport is a traveling of two solutes against each other, so against the path. So an example of an antiport is your sodium potassium pump. So right here is an example of a sodium glucose symport, where if we take a look at this behavior, we'll notice that the sodium concentration is much higher on the outside than on the inside of the cell. So this is the gut lumen versus the cytosol. Again, to pay attention to the direction. And then glucose in the cytosol is now going to get released into the gut lumen or the area of the gut. Okay. And we could see that originally the symport transporter protein is closed. Now, with both sodium and glucose present, what's going to happen is that it's going to have a higher affinity when both are present. It's going to bind together, outward opening, allowing both to come in. And then because glucose, okay, is using the energy from the sodium gradient, it's going to hitch a ride with the sodium ion coming in and having a much higher affinity, like I said. With that, the gate closes and then opens inwards. And so now you have sodium ions coming in with glucose going against their concentration gradient. So this is an example of a active transport in glucose case. Okay. So here again is the overall process. You have a sodium potassium pump, which we will talk about very soon. Bear with me here. Okay. Um, Wait, is this the sodium potassium pump In effect, yes, it is. No, glucose. It's look at glucose for now. Okay, so we'll talk about the sodium potassium pump in a bit. Okay. So there are three types of pumps. There's the P-type pump, the ABC transporter pump, the V-type pump, and the F-type pump. Know the difference between the four or the three. They're just going to be more of a definition base. So don't get too caught up on where we're going to find them. Just know the difference between them. Okay. And so that's pretty much all I really want you guys to know about this. All right. So going on. So now we're going to talk about the sodium potassium pump. Now, if we've had lecture already, and I believe we have, by the time you guys are viewing this, most likely there will be an in-class activity because this is something that makes more sense if you try to draw it out and kind of get through. So I recommend you guys who weren't at class during that time, again, I'm not too sure, but if you guys weren't at class and we did do an in-class activity, then what I would recommend is drawing out the sodium potassium pump. Look at what's involved in a nerve cell, how the gates are open, and then run through the steps. Now, there are a lot of YouTube videos out there that explains it in about 5, 6 minutes, and really that's all you really need. But remember, we're not looking at a physiological behavior in this concept. We do want to kind of apply it to receptor proteins, to pump proteins, concentration gradient, diffusion. So a lot of the YouTube videos out there will talk about, the action potential, the membrane potential and stuff. Yes, you need to know that. Yes, a lot of concepts overlap, but mainly get into the habit of looking at it from a protein perspective or a lipid, phospholipid perspective. Okay. So here we have one of these important pumps called the sodium potassium pump. Now, their main role is to pump sodium potassium against their concentration gradient. And this is mainly to restart or allow the membrane to get back to its original membrane potential and allow another nerve impulse to pass through, essentially. Okay. So we'll talk about this. I will draw it out, but just note that the sodium potassium pump is an example of a P-type ATPase, where sodium, three ions of sodium gets pumped out of the cell, and 2 ions of potassium gets pumped into the cell using phosphorylase or a phosphorylation um, from the ATP. Okay. Now, on the same lipid membrane, there's going to be a lot of different types of gates. They're going to be very selective. These are example of ion gates where depending on what ions being transported through, or there's sodium or potassium, it will pick and choose what it wants. Some gates are only specific to sodium. Other gates are specific to potassium, and they might have an obstacle in front right here. There might be a little, hole or a little protein tail blocking it. And sometimes, depending on the overall charge of the membrane, it might open or it might close. And that's fine. Okay. So here are some examples of voltage gated cells, ligand gated, intracellular, extracellular, and mechanically gated. Now, all I really want you guys to know is that depending on the membrane or depending on the ligand gates, what substance binds to it will either open or close it. Now, this is the end of chapter 11. I do want you guys to know the sodium potassium pump, but I'm going to go over chapter 14 very quickly. This is going to be a very quick PowerPoint, but I will be demonstrating the sodium potassium pump after this. So bear with me. Okay. So building on top of that, chapter 14 is all about the conversion, metabolic conversion in terms of the mitochondria or chloroplasts. Now, I don't really want you guys to focus too much on the cellular respiration cycle or the photosynthetic cycle. What I do want you guys to focus on is the concept of membrane potential. Now, does that mean that you need to completely forget what cellular respiration and photosynthesis is? No, because that is a gen bio background. You should already know cellular respiration, photosynthesis. Again, we had a couple chapters kind of covering that. But no, still know the background ideas of what photosynthesis is. It absorbs UV energy from light, gets into photosystem 2, photosystem 1, excites the electron in the chlorophyll pigments, and is using that excitation to run through the electron transport chain, having ATPase at the very end, pumping hydrogen ions from uh, in the inner membrane space of either the mitochondria or the chloroplast to outside or vice versa, depending on which process you're looking at. Okay. So again, because this is cell bio, we kind of need to pay attention to the transporters, the antiports, transports, symports type of enzymes involved. Okay. So again, I'm going to speed through this. The sodium glucose pump is important for this process. We are using this, yes, alpha ketoglutarate, malate, etc. Again, I'm not looking too heavily, too specific into these steps, but just have an idea that we are working with different types of transporters. Now, in terms of the mitochondrion, this is going to be slightly more specific just because it talks more about the hydrogen ion gradient, the electron transport chain. So, yes, you will need to know ATP synthase at least. Okay. So, ATP synthase, why are we using it? Where's the location found, etc. Okay. Outer membrane, inner membrane, again, how does this all fit in? That's essentially what I want you guys to really have an image about. Where's the ATP synthase found? Why are we using it? What type of transporter is involved? And how does the ions or the byproducts go in and out of the membrane? Because that's a chapter 10 concept, right? So if I'm looking at CO2, how does CO2 pass through the double membrane? Something like that. Okay. How does phosphate enter the mitochondria? So that's something that, again, is an example of what you would have an idea of. Okay. So that's the end of the PowerPoint. Now let's talk about the sodium potassium pump. Okay. So we're going to get started. I'm going to, again, chapter 11, chapter 14. What I want you guys to know is mainly the sodium potassium pump, but for chapter 14 specifically and what we're about to talk about, focus on lipid bilayer, concentration gradient, transport, proteins, receptor, ion gates involved, voltage gates involved, and so on and so forth. Okay, so let's get started. All right. So really quickly, I'm going to white out the screen. You guys can see that, I hope. Yes. Okay. And what I want you guys to do is kind of take a step back. So let's take a step back really quick and just take a look at where the sodium potassium pump is generally found. It's generally found in a nerve cell. And so I'm going to draw in blue right now. And I'm not going to draw the whole nerve cell. What I am going to draw is the axon, okay, as well as the next bit of the next nerve cell. Now, what I really want you guys to focus on right here is that we have at the axon tip or the axon terminal, we do have these pockets of vesicles right here that is ready to go when we need to. Now, inside these vesicles, we have things called neurotransmitters. Now, these neurotransmitters, okay, neurotransmitters can be a variety of molecules, acetylcholine being one, dopamine being another one, etc. And it will get released into this little synapse cleft right here. So we're just going to call this a synapse right here for just simplicity. But these neurotransmitters are going to get released in this region through exocytosis. Now, with the neurotransmitter being released, what's going to happen is that eventually one of the neurotransmitters is going to come into contact with a protein gate right here. Now, this protein gate will have a receptor attached to it so that when a neurotransmitter binds to this gate goes from a closed position to an open position. And I'm just going to draw open as that. Okay. Now, that is neurotransmitter reacting with acetylcholine receptors slash gates in general. Now, let's go on to the next thing. Once we have this understanding right here, okay, next question that we have to ask ourselves is why does the gate open? What is the purpose of opening this gate in the presence of a neurotransmitter? Well, if we take a look at the idea of what a nerve is, it's to send a signal. Now, our body is run through an electrical current or ionic gradient current. And so somehow opening this gate got to affect that type of electrical imbalance or electrical signaling. Okay. Now, before we get to the next slide, let's just set some ground rules here. Okay. I'm going to draw parallel lines to represent a lipid bilayer. Hopefully you guys understand that. We have embedded proteins throughout, but what we need to know is that on the outside of a cell, extracellular, in the cytosol will be the inside, cytosol. Okay. We know that there will eventually be a pump called a sodium potassium pump. Okay. Now, the definition of a pump is to pump something from an area of low concentration to an area of high concentration. This is active transport. So, sodium is going to get pumped outside to the extracellular space where 3 ions of sodium moves out and 2 ions of potassium, oops, oops, comes in. Okay, let me get my screen back up. And obviously it got to erase my image here, but that's okay. We're going to start on a new slide anyways. Okay, so we know, and I'm just going to say extracellular for EC and cytosol for this. And we know, okay, that we have the pump where 3 sodium goes out and 2 potassium comes in. So what does that mean about the location of the high and low concentration? Well, if 3 sodium goes out through that pump and 2 sodium comes in through that pump, that means we have high potassium, low sodium inside the cell, and high sodium and low potassium outside the cell. Okay, that makes sense, right? So if we open up all the gates, potassium will flow out, sodium will flow in because it's going with the concentration gradient. I hope that makes sense. Now, what does that say about the charge of the outside versus the inside of the cell? Well, if we're pumping 3 ions out and 2 ions in, and they're both positive charge, that means that we're going to have a stronger positive value on the outside of the cell than on the inside. So inside is relatively more negative than the outside. Okay. This causes a polar behavior, right? Where we have two poles or two different charges, one positive on the outside, one negative charge on the inside. So far, so good. All right. Now, because we have a higher positive charge on the outside and less positive charge on the inside, more negative, we have a thing called a membrane potential. Again, this is a potential energy that is caused if we open all the gates, it will spontaneously run through and give us some energy. And this resting membrane potential is roughly around negative 70 millivolts. Okay. So far, so good. Okay. I hope this makes sense so far. Now, I drew a couple proteins in here. I'm going to draw a couple more. Maybe we'll have a couple circles here. Fine. And then again, more plasma membrane. And then here's another box. Okay. So what is happening in the event that a neurotransmitter gets released and binds to a receptor protein? Well, what happens is that if it binds to, say, this receptor protein, this is your sodium gate, sodium ion gate. And because it's binding to a ligand or to another neurotransmitter, what's going to happen is that once it's bounded, this gate opens, allowing the influx of sodium ions to come in. Now remember, we have high concentration on the outside, so sodium is going to flow inwards. Now, as sodium flows inwards, what happens to your membrane potential? Well, we're resting at negative 70, right? Here's negative 70 to 0. We're resting at negative 70. If we have a whole bunch of sodium ions coming in, we're creating energy. There's a lot of energy being moved right now. So we are actually raising our membrane potential upwards. Okay. Now, negative 55, I believe, if I recall, is your threshold. So once negative 55 happens, there's a specific event that happens right here. Once we hit negative 55, because there is a change in voltage inside the membrane now, another gate opens up. This is your potassium voltage gate. And what's happening is that to rebalance out this process at negative 55 or something higher, again, I forget which one it is, but potassium is going to flow through this gate out because it's again at negative 55 something happens. Sodium is constantly flowing in, shooting your membrane potential up. And once potassium gate opens, then it shoots all the way down. But it's going to overshoot because everything is open now. Everything's equalizing. So we need a way to kind of cap these gates. So how do we do that? Well, attached to these proteins, we do have little plugs. One on the left side right here, one on this side right here. And once everything equalizes, these plugs are going to plug in the gate to close the gate back off or conform the gate into their shut position. And if that happens, and let me get my pen, the resting membrane potential is going to come back up and hopefully oscillate where negative 70 is. Okay. And that is, in essence, action potential trying to get recalibrated. Now, for it to completely get to negative 70, it needs to restart the gradient again. And this is where your sodium potassium pump comes into play. Because once everything equalizes, we got to reconcentrate both sides. So 3 sodium goes out, 2 potassium comes back in. Now remember, everything's shut at this point besides the sodium potassium pump. So what happens if we over concentrate the outside and over concentrate the inside? Well, you also have these other transporter protein. They're not transporter protein. These are channel proteins called leak channels, where in the event or in normal physiological state, we want to have these leak channels available so that potassium and sodium ions can flow through it to maintain its optimal concentration. Okay. It's like a sink, right? If you plug up a sink and turn on the faucet, the water is going to flow, but it won't overflow through the sink because there are leak channels embedded in the sink to prevent the overflow. And same idea here, we're going to need leak channels in the membrane to prevent the overflow or the over concentration of the sodium or potassium ions. But this is sodium potassium pump in a nutshell, the role of it and how it's being affected in the membrane itself. One last thing to remember is that this is a continuous membrane. A lot of students think that it's only in this segment that action potential occurs, but that's not the case. Remember, you have a continuous membrane all the way down. The action potential, the depolarization effect might happen in this section, but remember, as this one depolarizes and repolarizes, that repolarization or depolarization, right, is getting affected downstream and it's going to run the action potential downwards or upwards depending on the direction of impulse or the direction of the nerve. So, with that being said, it travels down like an electrical wire. Okay. So, in regards to what you guys need to know for these two chapters, it's a very short PowerPoint, like 15, 20 minutes, this PowerPoint. Proteins, lipids, gates, gradients, diffusion, transport, ATP, GTP, etc. Your book will go into slightly more detail. That's fair game on the exam, but in a nutshell, this is it. And again, if you guys were in lecture, we've done this activity in class together as groups. So for those who didn't come to lecture, I recommend doing this activity yourself. Okay? All right. Good luck. I'll see you guys in the next video.